So in this video we're picking up from where we left off on amino acids and proteins, and we're going to look at how amino acids turn into polypeptides, how they turn into proteins, and how the structures of proteins are formed through what we call primary, secondary and tertiary structures. So you all know, I would imagine, that proteins are very long chains. They're basically polymers. They're polymers of amino acids all joined together. They make up many parts of our foodstuff, our meat, eggs, nuts you can see on the board. Okay, so let's look at the polymerization of two amino acids to form what we call dipeptides. So we have, of course, a carboxylic acid group here, and we have an amine group here. And it is the OH group of the carboxylic acid group from here and a hydrogen from the amine group which allow for this polymerization to take place. Okay, so what you will see here, okay, is the remnants of one amino acid, and it has now joined to another amino acid, and that goes on and goes on really endlessly until the protein is made. Okay, so this is what we call a polypeptide, repeating units of amino acids joined together, and you get water produced for every single bond that forms. For every polypeptide bond, that forms, you get one water molecule. And this is a type of polymerization called condensation polymerization, where we make this small molecule, this being water in this instance. You now have a new bond called a peptide bond. So the peptide bond is the bond which links the one amino acid to another in the repeating unit, and very often you'll be asked to circle that or identify it one way or another. Okay, and there's your repeating unit. You see you've got three of them there on the screen from the amino acid, the general amino acid above. Now I can break this amino acid down by hydrolysis. So if I heat it with water, either an acid or alkaline, this is exactly what your stomach does with your proteins. It uses enzymes and acid and water to break these amino acids, these proteins, down into amino acids in your stomach as part of your digestion. The good thing is also that it means that proteins are biodegradable. So if an animal was to die in the wild, it will slowly biodegrade if it's not eaten by a predator prior to that. Okay. So in this question here, you've been asked to draw the structure of two dipeptides formed by the reaction of glycine and alanine. Now some of you might think, well, if I've got glycine and alanine, I'm only going to be able to make one dipeptide. Well, in actual fact, you're not. If you follow these two dipeptides through, you will get two different dipeptides. So a dipeptide is just two amino acids, not a repeating unit. We stop, if you notice, here, and we stop here. Okay, we don't continue the repeating unit on. Now, if you follow one of these through from one end to the other and the other one, you'll see that you have two different structures. And the reason why we get that is because in this one here, which is glycine, we have polymerized the NH2 group. And in this one here, which is our alanine, we have polymerized the COOH group. Then down here, what we've taken is we've taken alanine again, but this time for the alanine, instead of polymerizing the COH group, we've polymerized the NH2 group, and for the glycine here, instead of polymerizing the NH2 group, which we did up here, we've polymerized the carboxylic acid group. And believe it or not, you actually get two different structures here. So when you're asked about this in an exam, provided the amino acids are different, if the two amino acids are the same, you only get one potential structure. The amino acids are different. Make sure that you polymerize NH2 group of one with COH group of another up here, and then you flip them round and you do the others down below, and that will give you two answers, as opposed to most people think initially before you practice these that you only get one. Okay, so have a go at this one, which is 511E, please. To pause the video and have a go at that. Okay. Now this question here popped up, gave a lot of difficulties. Most people were able to get the two dipeptides. So the two dipeptides, okay, were polymerization of that with that. Okay, so we got H2N, CH3, H, and then we polymerized our group there with, as I said here, Okay, CH2OH, and then H, and we had our COOH left over. And then we flipped the molecules, 180 and 180, both of them. Okay, and so we had HC, or HOOC, sorry. Okay, CH3, H, this time I'm polymerizing the NH2 group of this one with the COOH group of the other one on the right hand side. Okay, and we end up with 2 dipeptides. Now a lot of people, then the question is, and one other possible repeating unit, which caused all sorts of difficulties to students during this question. Well, the other possible repeating unit was you had a cutting on to the fact that you had an alcohol group here, and with the alcohol group you had a carboxylic acid group here, and if you remember, that forms an ester. So therefore your final answer, which very few people actually got, was an ester functional group. Okay. So the ester was CH2 there. See, we have H there. We have COOH there. And we have H there. So there's our ester functional group there. So that was the third repeating unit. You got a mark for that, a mark for that, and a mark for that. Most people got the 2 in the red on the left hand side. Very few people got the ester group on the right hand side. Okay, again, you can pause the video, have a go at the ones that I've put on the board there. So you have a couple of dipeptides which you should be able to work out. So in actual fact, the answers to those are these 2 here. So we'll just clear those and you can check your answers to that question are the same as what's on the board now. Okay, draw the structure of a dipeptide formed from 2 molecules of this amino acid. So it's just one dipeptide. When you've got just one amino acid reacting with itself, you're only going to be able to get one possible dipeptide. So I would do it like so. CH, this part of the molecule here remains unchanged. This is the part which will polymerize. I line it up with another one, the NH2 group of it, of course. Okay, and again, this part of the molecule I just redraw down there, and I end up with the COOH there, and that was the dipeptide that was formed. So again, check your answer. Okay, have a go at this one. This is exercise 511E we're on to now. Check your answers. Okay. Right, peptide bonds, as I said earlier, are hydrolyzable, which means that they can break, they can biodegrade. It also means that they can be digested in humans, in our stomachs, and animals as well, of course. Okay, now we can speed that process up in the lab if we wanted to by heating it under reflux with concentrated hydrochloric acid, and we can use then paper chromatography to actually identify the amino acids that a particular polypeptide are made of. Okay, so we can either break that protein up completely and identify the individual amino acids, or we can carry out partial hydrolysis and work out the dipeptides and the sort of tripeptides that are being used or that are sequencing together to make particular amino acids. Okay, and that will allow us to work out what's called the primary structure of the polypeptide. In other words, the sequence in which the amino acids come in that polypeptide or protein structure, like so. Okay, so amino acid 1, 2, 4, 3, 3, 2, etc. And that's the primary structure of an amino acid. It's just the sequence in which those come in, those amino acids come in. Okay, so polypeptides or proteins have a fixed sequence, so a particular protein has a fixed sequence of amino acids. Those then have the ability to coil and to fold based on intermolecular forces within that primary structure, and that gives us the secondary structure. Okay, you have 2 types of secondary structure you need to know about. One's called an alpha helix, one's called a beta pleated sheet. You don't need to know much more than that, okay, but they are formed, okay, by internal hydrogen bonding. Now they are formed from the remnants, okay, of the C double O, okay, in your protein structure and the interaction of other NHs from your polymerization. So when we were drawing our dipeptides there a couple of minutes ago, and we were seeing in our peptide link we had this. So the COO hydrogen bond with an NH from a different amino acid residue in your protein, and that gives us this folding and coiling to give us our alpha helix and our beta pleated sheets. That structure then can fold upon itself further and bend upon itself further by using interactions, okay, to give us the 3D shape of a protein. So a lot of you who do biology will be thinking sort of enzymes now and shapes of active sites. This is caused by van der Waals forces, ionic linkages, hydrogen bonds and disulfide bridges. Importantly though, this is now between the R group. So up here we had hydrogen bonds between the COO and the NH of different amino acids. Okay, now this is caused by interactions between the R groups of different amino acids. Okay, and those three different things are primary, secondary, tertiary, and you will need to know those. So I've kind of given a bit of a diagram here of it. Okay, primary, simply the sequence of the amino acids in the protein. Secondary, and it's probably a little bit hard to see here with the colour of another diagram. There's the COO of one amino acid and the NH of another, and you can see we've tried to show there the dotted line, the hydrogen bond in it, and the same over here. There's an NH of one amino acid and the COO of another, and again there's the hydrogen bond, which allows for the secondary structure, the alpha helix or the beta pleated sheet. Beyond that then, if we were to put the R groups in here, the R groups then they start to interact with each other, and that's what gives us our secondary structure. Quaternary structure is our tertiary structure. Okay, so I think I've tried to show this in the secondary structure, the alpha helix here, in a little bit more detail if we can. So maybe you can see there's the COO, okay, and there's an NH, there's a hydrogen there, and there's our hydrogen bond in our secondary structure. And that results in an alpha, in this case an alpha helix. A beta pleated sheet, same sort of idea. I've tried to show it here with these yellow lines. There's your COO, there's your NH there, and then we've put in the yellow bar to show the hydrogen bond, making the alpha, the beta pleated sheet, sorry, of which is a secondary structure. Tertiary structures then are to do with R groups and interactions of R groups. You can see here we've an R group, okay, forming here van der Waals forces between one R group and another. Down here we have hydrogen bonds between one R group and another, so these form the tertiary structure. We have disulfide bridges between R groups here, and down here then another type of interaction of R groups are ionic bonds. And you can see, guys, why proteins might start to unravel. So if I heated this protein, if I increase the temperature, these very weak forces will start to unravel, will start to break, and that causes the protein to lose its shape. And once the protein loses its shape, it can't do what it's supposed to do anymore. Also, if I change the pH of this, you can probably see where some of these molecules or some of these structures, if I put this into, let's say, alkali, there's a carboxylic acid group that will become deprotonated to a COO minus. So that will affect the hydrogen bonding here, and if that affects the hydrogen bonding, then of course that's going to affect the shape of the tertiary structure. So changing pH on enzymes in particular has a significant effect. If I put hydrogen ions in, if I made it acidic, hydrogen ions will probably protonate that, turn it into COH group, and destroy this bonding here, this structure. Okay, so you can see how both change in temperature can cause the tertiary structure to unravel, and change in pH can cause the tertiary structure to unravel. Okay, so exercise 511F, you can pause the video and have a go at that, or go to your textbook and do it. Okay, again, pause the video. Again, pause the video. Don't on this one spend too long, pick out the key points of primary, secondary and tertiary. Don't be writing an essay on it, okay? Okay, again, pause the video. Okay, so that deals with primary, secondary and tertiary structure, guys, of proteins. We'll leave it there. We'll come back to how enzymes are made and how enzymes are work in the next video.